I'm the head of discovery informatics at e-therapeutics. We're a small drug discovery company based in Oxford in the UK, and as Terry alluded to, what we do differently, what our niche is that all of what we do is data-driven or computational based, at least in the early stages. What I wanted to go through today wasn't necessarily specifically what we do, although I do cover this. I wanted to give a bit of a broader sort of overview of some of the computational data-driven approaches that are going on in our industry at the moment, because it's going through a bit of a digital transformation, I guess, if you want to use the hype term, and there's some interesting stuff going on. So, there we go. Before I get into the details, I want to give some motivation, because I sort of realize that most people here probably don't know much about our industry. So, it's basically an industry that's ripe for innovation at the moment. This is a graph that came from a paper from 2012 looking at the productivity of the drug discovery industry. And this graph is one way of measuring productivity. It's basically the number of drugs approved per $1 billion over time. The y-axis is logarithmic, so that's an exponential decrease. The author of the paper named this E-Room's law. It's Moore's law in reverse. Basically, our industry is getting worse and worse in terms of its productivity over time. And this is in the face of novel technologies, post-genome, post-sequencing of the human genome, all of the genetic engineering technologies, etc., don't appear to be making much of a difference in terms of our productivity. This graph shows a similar thing, but from a different point of view. If you concentrate on the graphs or the bars on the right there, which show effectively the total cost in millions of dollars of getting one drug to approval, and the different bars are different decades. So what you can see again is an exponential increase there. I don't know if it's truly exponential, but it appears to be an exponential increase in the cost of getting a drug to approval. The last bar in black there was from last decade, 2000 to 2010. We're at $2.5 billion now to get one drug through to approval. So I think both of these graphs show there's a massive need for innovation in our industry to increase productivity. The other aspect of my clicker's not working. There we go. The other aspect of innovation is more, what are we trying to do? And that's improve human health. These two figures that I found from various reports try and get at some of the underlying issues there. One of the biggest ones is that the world's population is both growing and aging. This graph shows the change in the mortality rates due to communicable nutritional diseases relative to non-communicable diseases. And basically the world is moving to dying from non-communicable diseases. We're getting older, we die of cancer, etc. now, not of nutritional, communicable diseases of the past. And the graph on the right-hand side shows one of the key, I think, issues that's going to face society is facing at the moment and will continue to, and that is the increase in dementia and neurodegeneration in general. This is a projection on the top there of the number of cases of people with dementia over time, and then the bottom is the forecasted cost on global society. And for us as an industry, I think this is one of the biggest issues that we're facing. We are now at the point, if you take Alzheimer's as one of the forms of dementia, every single drug now that has been tried for Alzheimer's has failed. We are no closer to success for Alzheimer's than we were, how many years ago. And I think that is pretty depressing, to say the truth. So I think, there's some productivity needs from the business point of view and some big health needs that our industry can use some novel approaches. Again, because I'm sort of aware of the audience, I wanted to just give you a gross overview of the drug discovery process. As you can see, what this figure tries to illustrate is that the drug discovery process is basically one big continuous set of filter after filter after filter. We start on the left-hand side there with, and these numbers are sort of small, it's more like 1 million compounds quite often rather than 10,000 that are tested generally in the lab to find things that look interesting, and then they get taken effectively through more and more aggressive filters, both preclinically, are they safe to put into humans, and then into the clinic where they're tested usually against healthy volunteers to see if they're toxic in humans, and then through wider and wider, more heterogeneous populations until a drug is approved. The timelines there are actually, again, this graph is, I need a better one. This graph is underestimating some of these timelines. It can be a lot longer than 10, 15 years, especially in some of the cases like Alzheimer's disease. And you can see some of the numbers here illustrate where the costs come from. Late-stage clinical trials, thousands of human volunteers who go into those trials, very expensive. Failures that happen in the late stages are extremely expensive, and there's a lot of evidence now that a lot of the failures that are happening in the late expensive phases are occurring because the things we've discovered in the early stages just aren't doing what we thought they were doing. And again, a lot of the innovation that I'm going to talk about, the in silico innovation, is going on to address those issues. So, why is this? There we go. I'm going to talk about the first early stage that I've highlighted here, the preclinical stages of drug discovery, and in particular three areas that I'll go into in more detail. This isn't to say that there isn't anything going on in the computational data-driven approaches on the clinical side. There's a lot going on there, some very interesting stuff. I just don't work in the clinical side, and I've only got a short amount of time. So I'm going to focus on the preclinical side. So ADMET. So the T in ADMET is toxicology, which is the basic question of, is a compound safe to administer to humans? Computationally, what does that mean? You have a compound structure, the chemical structure of the potential drug. You want to know, is it going to be toxic? That's the model in question. There are multiple potential mechanisms of toxicity. Does it cause cancer? Genotoxicity, does it damage your DNA? Reproductive toxicology, anyone who's old enough to remember thalidomide will understand where that comes from. So all of these aspects of toxicology have to be addressed before the regulators allow you to put it into humans. And to illustrate the success of some computational data-driven approaches, I was going to mention this company LASA, which is based out of Leeds in the UK, and they have now multiple products on the market that use computational approaches for prediction of toxicology and other ADME properties actually as well. Derek and Sarah, they're probably their two well-known products. Derek is their older product, which is actually an expert system, knowledge-based system, where they actually convinced a number of the bigger drug companies to actually give their data to them and acted as a sort of honest broker in the middle to build that expert system out, and then give the predictions back without sort of releasing that proprietary data back. So that was what they figured out how to do. And now with the Sarah product, they're doing it in a statistical machine learning modeling approach as well. And I think very interestingly, and this is the only example I know of this, but there may be others, the outputs of their computational modeling, or one particular one actually, are now acceptable by the FDA in regulatory submissions instead of doing animal experiments, and which, of course, the productivity savings of that, etc., are pretty large. Just to sort of nod to the open source world, we have something similar in our industry, although nowhere near as big. There's a project, OpenRiskNet, which is actually a sort of similarly motivated project, looking at toxicology, specifically around the data sharing issue that I mentioned, and also about sharing predictive toxicology models. It's quite a new project, and I'm sort of keen to see where this goes. ADME is basically, the way I put ADME is all of the properties, of what makes a good drug, except for does it work and is it toxic. So it's basically, you know, how does it get into the body, how does it get removed from the body, etc. And there's a whole field of, in our area called quantitative systems pharmacology, which is all based around the modeling of drug distribution in the body via systems of coupled differential equations. Big history to this field over the last couple of decades, and it's actually been pretty successful. It's routinely used now. LASA, again, has one product around metabolism prediction as well, but I'm not going to go into that in much more detail. So that's it for ADME. The next aspect of preclinical drug discovery I want to mention, where computational approaches are having actually a very big inroad in the last 4 or 5 years, and that's around medicinal chemistry. So medicinal chemistry is the process of taking your initial, what we would call a hit in our world, potentially interesting compound, generally won't have all of the properties that make a good drug, and they are chemically optimized to actually make them better in multiple aspects. So efficacy, does it work? Can we make it work better? So we have to give less of a drug to a patient. All the ADME properties I just mentioned, and also IP issues as well. Where am I supposed to be pointing this? There we go. This is inherently, if you think about it from a computational point of view, a multi-objective optimization problem. There are multiple objectives there that we're trying to optimize. And critically, these objectives, these properties, are actually quite often conflicting. You know, we tend to call it the sort of whack-a-mole problem. You increase efficacy, something else gets worse. And humans are terrible at optimizing multiple objectives at the same time. It's just we are. So there's a lot of data-driven approaches that have been attacking this problem, thinking about and designing systems to actually help in this multi-objective optimization problem. And these typically model the structure-activity relationship either from a statistical point of view or from a mechanistic point of view, or both. And what they're generally doing is an active learning approach. So compounds go through a design, make, test cycle. You design a new compound, you make it, you physically synthesize it, and then you test if it's what its properties are. And you carry on around this cycle until you optimize. The data-driven approaches now are taking that design piece, what compounds shall I make, and predicting them now from computer modeling, and then taking the results from the test in the lab and then feeding back into the models and learning as the process goes. And there's a number of companies, these are the 3 that I know of the most, that have been very successful in this, taking 5-6 year project timelines down to a year, for example, in terms of productivity increase. And I would say this is, in our industry right now, at least preclinically, the best example of where the application of data-driven machine learning, AI, whatever you want to call it, approaches to the improvement of productivity in the industry. Now I wanted to get into the final piece, which is biology, which is where e-therapeutics operates. This slide attempts to try and illustrate what the key issue around drug discovery is in terms of the biology. Phenotype, which is the term we use for behavior effectively, disease phenotype in particular, is a complex property. It isn't a proper property of individual molecules. Cells are made up of multiple molecules, tens of thousands, and they interact together in networks, like Facebook networks, whatever. It's the fact that they interact in a complex network that gives rise to behavior. Disease arises because you get dysregulation of normal networks and disease networks occur within the diseased cell. However, when you're designing a drug, you have to intervene on an individual protein. Generally, a compound will bind physically to one molecule in the cell. So there's a gap there between the individual molecule and the phenotype, which is driven by the interactions of thousands of molecules. So the question is, how do you significantly perturb those networks that drive disease biology by intervening on individual molecules? This is the biggest problem, or the big problem, that underlies all of early stage discovery. I think it's one of the problems that underlies why the fact we haven't discovered any drugs for Alzheimer's. It's a very complex disease, etc. So we as a company are taking a computational approach to this. Some of the, the points are illustrated there of why this is an interesting thing to do. Lab techniques are inherently reductionist by their very nature. They have to be. They don't bridge levels very well, and combinatorics kills laboratory experiments. Computational approaches can address these issues, and that's what we're trying to do. So rather than try and describe the maths and the computational algorithms that we do, I thought I'd illustrate it with a couple of videos. So these pictures are of cellular networks from a particular disease. I believe it was cancer. One aspect of what causes cancer from a drug discovery project we ran about 4 years ago. The red blobs are proteins, they're molecules within the cell. The blue lines are known interactions. These proteins talk to each other within the disease state, within cancer. There's an inherent property of biological networks in general, and that is that they're robust to perturbations. I've just labeled 10, 15, I believe, nodes, proteins within that network with those green labels, that in a second they'll be removed from the network. They're gone. That network's been perturbed quite significantly. Not much happened to that network. It's robust to that random perturbation. I should have said I picked those nodes at random. It's that robustness property that is thought to underlie biological robustness in general. We are robust to all the random changes that occur to our genome, etc. We die otherwise. Evolution has made us robust. And it's the fact that behavior arises from networks, not individual agents within that networks, that gives rise to that robustness. However, the other side of that robustness property is if you find the right set of nodes, you can have actually quite large synergistic effects on that network. I've just labeled the same number of nodes, not randomly this time, but using the analytics we've developed in-house, and in a second they'll be removed. And you can see here something like dramatically different happens to that network. Its core structure is completely destroyed. As I mentioned, this network, we believe, is what gives rise, in this case, to the properties of cancer cells. What we are looking for in our drug discovery process are computationally compounds and can give rise to the effects you saw on the right-hand side there in the second video, whereas the vast majority of things will do what you saw on the left-hand side. So that's illustrated in the video there, basically the whole approach to our drug discovery process. Game on. I'm not going to go through, this is a sort of idealized linear flow of how we approach drug discovery. I'm not going to go through this in any detail from a biological point of view, but I wanted to illustrate the various computational in-memory computing approaches we use in the three distinct aspects of our process. So the first one is, how do we construct those networks that I just showed you in the previous slide that represent, for example, a mechanism within cancer? This construction or reconstruction is effectively a reverse engineering approach. You take measurements from diseased cells and you try and reconstruct the underlying mechanistic model of what was happening in the cell you just took the measurements from. The issues, there's a lot of noise in biological data. There's a lot of experimental bias in the data we can get hold of from the outside world if we can't do the experiments ourselves. The computational techniques we generally use here are optimization approaches around linear programming. And on top of that, a lot of statistical modeling to account for the various biases and noisy data. So a lot of distributed parallel computing in that aspect. Not particularly data heavy. The sets we're talking about here aren't very large. The middle stage, how do we identify those significant intervention patterns I showed you on the video on the right-hand side? Computational techniques we use here are various techniques from network science and network analytics. You're all aware of PageRank. There is a whole mathematical field around similar things. we use various approaches from that, and a lot of stochastic optimization approaches, such as genetic algorithms, to address the combinatorics that we're trying to address, that we're trying to overcome. Again, not particularly data heavy in this side, but a lot of distributed parallel computing. Genetic algorithms, for example, are very parallelizable. They're embarrassingly parallelizable population-based algorithms. By utilizing the distributed computing, both of the techniques we use in these first two stages become feasible. We wouldn't be able to approach drug discovery from this point of view without that aspect of in-memory computing. Then the final piece, compound mapping. So the question here is, okay, we've identified the intervention patterns. There's sets of proteins I showed you in the video that can bust apart the network. Can we find compounds that can give rise to this? This is very data heavy now. We have a database of about 15 million compound structures and their associated biological effect, which proteins within the cell they can modulate the activity of. That's billions of data points. Even with all of those data points, it's still pretty sparse. Again, there's a lot of experimental bias in that data. Excuse me. So we use machine learning predictive approaches quite aggressively on this data set. And there's a couple that I've put there that we've been successful with, matrix completion and gradient boosting, if any of you come from that world. And again, on this part, we use in-memory computing. Quite interestingly, we first started down this, how do we approach the machine learning on this? The usual way you would do it, you take your massive data set, you subsample it to a representative set, and you do your data science on that small set. It didn't work. We couldn't find a good way to subsample that data set down without introducing biases that the model then captured. So we gave up on subsampling, and we basically do all of our data science on the full data set. For that, we need a distributed grid. Again, I'm not going to go through this in detail. This is just to show that we have now run our approach on a number of different drug discovery projects in a number of different areas: oncology, immunology, immuno-oncology, CNS. And what the graphs on the right-hand side show are just some summary statistics on two things. The top one is how many compounds do we actually after our in silico phase, we take the compounds into the lab and test them. How many do we take into the lab and test? And on average, relative to what normally goes on in our industry, we're testing 3 orders of magnitude less compounds in the lab. We test 300 to 500, 300 to 1,000. The typical in the lab is, as I say, 3 orders of magnitude higher than that. Obvious productivity and speed aspects to that. The graph on the bottom shows what is our success rate. I should say our numbers are in pink. We are getting a success rate of 2 to 3 orders of magnitude higher than the average success rate from normal approaches in the industry. Again, another indicator that I think what we're doing is useful. Maybe the more interesting thing is I come back to the Alzheimer's thing. There's a trend in our industry now that's sort of realizing that one of the big reasons that things like Alzheimer's and other diseases are failing late on is that the things we test them, the assays we test them in initially are too simple and they're not representative of what happens in the human system. So there's a move now to use preclinically more and more complex cellular-based assays in the test tube or in the lab. The problem with those is that they're not industrializable. You can't use the techniques that have been developed in our lab of lots of robots screening millions of compounds against these complex assays. They just don't miniaturize enough. So you need techniques to effectively do, we believe, in silico screening, to take that 15 million that I mentioned we have in our database, come down to a smaller number, 300 to 1,000, that you can then go into the lab and test. So this combination of in silico computational screening followed by screening in the lab with way more complex assays that are more representative of the human system should give rise to drugs that are more likely to affect the human complex diseases. Just to summarize, I think I showed you some motivation of our industry is actually ripe for innovation at the moment, and we are certainly going through an intense period of active exploration of data-driven approaches. I'm a little worried we're at the top of the hype curve and it's going to crash soon. We'll see where it goes. Hopefully, some of the good things will stay around, and we won't just all get thrown out with the crash. There are numerous aspects of the discovery and development pipeline that can benefit. I've not talked about clinical development. There is a lot of good stuff going on there as well. And I've gone through 3 examples of what's happening in the early discovery case. And I think the key here is the potential benefits to both patients and society in general. Reducing the cost by increasing the efficiency of the discovery process. There's a lot of noise at the moment about how expensive, especially in the US, certain drugs are. A lot of reasons for that, some good, some bad. Increasing productivity has got to help there. And I think the other aspect is the aspect I mentioned around Alzheimer's and neurodegeneration and other diseases similarly. There are some serious unmet medical needs that we've effectively failed to do anything about. And I think novel approaches has the potential to start addressing those. And that's all I wanted to say. Thank you.